Hello, Ujjwal.
Hello, hi sir. How are you?
All right, how are you?
Yes, sir, fine. So actually today, I think I won't be able to show CC 3D progress. I don't know why. Like the software has been recently crashed. I'm working on it to recover it.
Okay.
But as soon as the 3D model is, I have some work. I have done some work for the in the Photoshop for 3D models. So I will like take 5 to 7 minutes to show that.
Okay. Sounds good. Why don't we wait for a few more people and then we'll start.
Okay, sir.
Hello, Jesse.
Hey, I won't be able to really talk much, but I'll be listening today.
Okay.
How are you?
You're at the conference?
Yeah, I'm sort of at the head. I typically have work now anyway, so yeah.
Okay. Hello, Dick.
Oh.
All right. Okay. What's going on in your end, Dick? Anything?
What's going on? After we're done, I'm going fishing.
Okay. That's good, I guess.
Yeah, I'm going to eat off the land.
Yeah. Okay. All right. I guess we can get started. I don't know how many other people will show up, but they can come in. So, welcome to the meeting. Hope you've been having a good week, or more than a week if I haven't seen you since. Jesse, welcome back. Jesse's at a conference today for machine learning, so he's doing some live tweeting of that and other things. So, and then Dick, I got an email from you about something yesterday. It was a book.
Yeah, this guy's producing a book on called Biological Physics.
Okay.
Okay.
Yeah.
Anyway.
Yeah.
If and when the Embryo Physics course is available, send them a link.
Okay. Yeah.
Okay.
Yeah. We'll talk about that today, in fact.
Oh, okay. Okay, good.
And then Ujjwal is going to present something in a bit. Why don't we start with Ujjwal's update, if he's ready?
Yeah, just
yeah, we can just share screen share here. So this is his he's an update on some 3D modeling work he's doing.
So I'm working on the model for this C.elegant Embryo, and I have taken the labeled movie from the OSF website. That movie is yeah. So this is the movie that I am working on. So this shows labeled diagram of how the C.elegant embryo is being divided. So this is the diagram. So I am just taking off the number of frames from this movie, and after that I am producing this type of models. So these type of models are created in sequence wise, and I am giving a transition between them so that you can stop at every point, and this allows a specific kind of flexibility now. You can have a 3D view where you can see the whole model in 3D, as well as you can adjust parameters you can say yeah. So you can definitely adjust live models, and the best part about this is like we can also embed them in websites. So what I'm trying to do is create a Woo frame-wise sequence for these kind of uh, for this model, and then do some animation so that they can be emerged simultaneous. How it is different from video is like you can zoom it out. You can also zoom in and zoom out these models in website as well, so you can see the each and every segmentation page very clearly and labelings. And also, like when we have started the project for GSOM, and then, like I have seen the winner has given some good distinct labeling for the cell segmentation, but it is also like we can say like still in the early phase. So like once we have got the very good model, we can also incorporate it with the here so that we can create real-time segmentation, something like that, uh, model so that more and more people can see. And also, I was working on these models and seeing like we can also import some sort of PhDs in Python programs so that more and more people who are using DevoWorm library, we can include these 3D modelings in the Python program, so they can run models and compare in the real time. Also, like I have been working on CC3D, so like the software is crashed somehow. Like I'm working on recovering it, but what I have done till now in CC3D is like I have made a program for cell sorting first, and then the condensing and non-condensing cells I have provided the parameters so that non-condensing and condensing cells attract much. And after that, I have seen like I have given some parameters, virtual touch and color, etc. So like I was near about to make model for the growth of C dot element embryo, but the point is like in the CC3D you can't have that some this sort of label diagrams there. Like it is some simulations with different color. So I was also studying a paper about I think Cylon and Novak cell time lapse, something like that, where they shows like how the cells are being grown in a cell clock. So I was working based on that paper. So I think I have to present that today, but somehow my CCP did crash.
Oh, yeah.
So I think I will be able to present it in future meetings. So the first task which I am doing right now is creating the animation so that we can get this model uh, completely, and we can embed this in the website itself and in the library so that people can compare side by side. And then I will see like if I have to restart my work on CC3 or somehow I will able to recover it.
Okay. I have a question in the chat. Uh, Richard says, "Are the segmentations 3D?"
Yes, like right now it is just I have taken the image and do some intrusion work, extrusion work on the image. But yes, if once we have the segmentation clearly labeled, like once we have the model for segmentation, we can definitely have a different 3D segmentation, and we can also set that. I'm also working on something so that we can set them these cells can be set apart different entirely, so that we can focus on each of the cell and their diameter. We can work on diameter either on Python, or we can also work on getting diameter of each cell in the software itself.
That's good. So yeah, so this is one slice of an embryo. So when they image an embryo, they image the embryo in slices. So you go from like the top to the bottom of the embryo, and you take like a slice, a focal plane slice, of the embryo. So this is like the middle, I guess, of the embryo. These movies that you have, they're rather old movies, so they're not like the highest level of technology available today, but I think they're still pretty good videos because they have the whole sequence, and it's the entire. It's sort of the entire embryo, all the cells at any given stage. So I think that's a good place to do this. Yeah, I think you can't extrude from this image though, so we can have like we can take that one flat focal plane and we can stretch it out into cells. I mean, we can approximate them.
Yes, sir.
Yeah, and so then we'd have a model from that. And then CC3D you're working on, is that model like sort of joined to this model, or is it something separate? I'm not quite sure. I understand.
Oh, I was like, I can give the link of the paper. I was studying this paper. I think this one. Like I will provide the link in the description in the Slack channel. So I was they are something like Cyclo and Novak theorem for the cell growth. So what they are doing is, if I can show some YouTube, they also have a YouTube video, I guess. So I don't know what its name is. Yeah, yeah.
Look at that one.
Yeah. So this one is the video on which I am working. Basically, this is uh, it is a paper written on the cycle model for Tyson and Nova. Sorry, I am mispronounced it. So it shows like how the cells are being grown in the period of cell time. So they are talking about some cell time, and as if I can share, so like this is how the cell is being grown. So I think like I found some similarity between this type of growth and what embryo is being grown. C. Elegant embryo is grown. So I'm working with this paper and I'm trying to see like if we can convert. So yeah, I have done some work and it looks pretty good. Like I don't have to show this right now, but yeah, in the future meeting I will definitely show. This is how something I'm working on.
Well, that's good. Yeah, this looks pretty good. Could you put a link to that YouTube video in the chat? Yeah, so this is the three copy cell 3D model. Has been quite elusive to the group over the years because I visited them in like 2015, and we haven't really been able to do much with it since. Just there's a learning curve, and then you have to find the right person to really kind of dig into it and build some models. But the models are kind of like what Ujjwal was showing in the video. They're sort of agent-based models. You can model cells, and you have this field of cells, and it's you run a simulation. You give the cells properties, and you can run simulations of cells over some process. They have a lot of models for development, not necessarily C. elegans development, but like chick embryo development, and there are a couple other models of development that I don't know. Maybe they have it on the YouTube channel there, but it's a pretty versatile program. So I like this. So this is good. Good progress. Again, I think the next, I think your next steps kind of. You've kind of mentioned those. So you said that you were going to sort of work this into a 3D model. You would have, you could do this, and actually you could do this template for any number of videos. So this is just one example. There are actually there's data captured where people have, many focal planes through the embryo or through the tissue that they're working with. And so you can actually do the same sort of methodology for those sorts of data sets as well, where you have, images at different focal planes, and you can, put them in a model like this and then extrude them out. And in fact, in that case, you could use the information from each slice to extrude in some way, in some more educated way. I mean, you can extrude from this pretty well, but I don't think there's a lot of variation on the dorsal ventral axis, which is the axis where you're going from the top to the bottom of the cell. But you do have a lot of variation, of course, along these two axes that you have in this image. So this is where all the cells divide. It says how many slices.
Right now, like from a video of 31 seconds, I make around 230 slices, 230 frames, and I'm targeting to make all 230 major slices so that this video transition, animation transition will be smooth enough, so it don't get it doesn't feel like it is something like we have connected two images between the transition. It becomes smooth. So yeah, around 230 frames.
Okay. I think that's the right. I think that answered your question. So yeah. Well, that looks good. And then you'll be giving further updates, like in coming weeks. And so yeah, keep us posted on that.
Yes, for sure.
Okay. Next thing I wanted to do is go to the. So I actually told Dick that he knows about that. I've been working on getting the old Embryo Physics course into a new format, and so I will be showing that now. So this is the home of the new Embryo Physics course, open course materials based on the Embryo Physics lecture series. Then, hosted 2009 to 2014 in Silverbog Second Life. So for the people who weren't there, of course, which is Jesse and Ujjwal, Dick put together an embryo physics course. And this was just a number of topics in embryology and development. And he brought in people from all over. People brought them in virtually into Second Life and had them give these talks. So there's a lot of content here. There's something like, probably about 15 talks a year. So that was over 5 years. So that's a lot of content. That's and it's been hosted on someone's blog, so it's online now. But we wanted a little bit more permanent home for it. So the first step in that, of course, is to put it in some sort of archival location instead of someone's personal website. And so for this right now, we're using GitHub. And so GitHub is a secure server, and it's a permanent server. I mean, GitHub is a pretty popular brand. It's owned by Microsoft, so it's not going to go away anytime soon. Once this is finished, this can be archived to something like Zenodo. And so the benefit of that is to have like zip files that people can download, and it would have like a DOI on it. So it would have like a more permanent address for people to cite it or things like that in papers. And so the way this archive is set up right now, there are a couple folders here for different. This is media, like images. These are movies related to the lectures. The presentations, which are the PDFs of the lectures, and then some reference lists that get linked in the main documents. So these are the main documents here, or the main sort of presentation locations. So when you want to pick a certain year, so you want to find out about lectures from 2009, you can go to the 2009 lectures document, and you see that there's a header here. So this is what it looked like in Second Life in 2009. I had some old photos that I used as headers for these years. And so this is an example of Silverbog. Oh, there we go. So the way it was set up was that you'd go in, and here's some seating for the avatars. You would go into Second Life and choose an avatar. So this was my avatar at the time. And you'd go into this location in Second Life, and your avatars could sit around this campfire. And then here are the boards. So these boards were where you would watch the presentation. So here are a couple of the presentations. These are PowerPoint slides projected onto these boards. And then you would watch the board. So you could start this presentation here at the start button. And you could walk through the presentation with these arrows. So you could go forward and you can go backward. So anyone could look at some of these presentations that are put up on these boards. They're kind of like there for visitors, anytime if they came through the place. But then when there was actually the lecture, at the time of the lecture, you'd bring up that week's lecture on this board, say, and then the whole group could sit there and watch it. And then the master of ceremonies would advance the slides, and it would be, a formal lecture. But you could also visit, in the off hours and view any one of these slideshows. So that's how it was set up. And right now, what I have here is this. So this image just kind of orients people to this, because people probably don't know what Second Life is, what was going on. We might have some more visual aids here if Dick has anything that he'd like to put up as some additional media, that would be great. But this is what we have right now. And so then we walk through the schedule for each year. So this is 2009. In 2009, we had weekly talks starting January 29th. And so we have Stuart A. Newman from New York Medical College. And he gave a talk, A Pattern Language for Animal Form. And this is the abstract. And then you have links to the slides and to a related blog post. So the slides, this is the link to the slides. These are all hosted on this GitHub repository. You can click on this. I don't know if you can actually see the slides or if you have to download them. Okay, I think it's going to bring up the slides. But you can download this once it's open. People click on the link, they can get access to this PDF. And then download it. And then, view it on their own machine, or you can view it in here. I don't know if it advances in GitHub. But in any case, you have access. It's archived in a static location. And so, each week we'd have different speakers. This was Bruce Damer. He did a virtual world presentation. So this wasn't development per se, but this was about virtual worlds. And so that's another talk. We have some talks that don't have abstracts. So some of them don't have abstracts. You just have the slides. Most of them do. And again, there are links off-site, so related blog posts, related presentations. Actually, these related presentations are talks that this person gave in another year. So, there's sort of a hyperlinkage between their talks from different years. So if I'm interested in this talk, I can see other talks. And I think it was set up like that in the old archive, but I brought that forward. And so in this case, we have someone who was presenting something that was very movie intensive. So the talk relied, a lot of developmental biology lectures will rely on movies because, as you've seen, that's one of the main forms of data. And so in this case, we have the movies, and they're sort of in this area where the abstract normally is. So we just have the links to the movies. And those movies are actually on the local server as well, on the local archive. So they're all sort of, they won't go away. And yeah, so that's the way that's set up. And so that's basically, I'm not finished. I don't know if I'm finished with this year yet. I think I'm finished with this year. But there are a couple years I still have to go on this where, I have to bring over the abstracts and the links to the slides. So 2013 is another example. So this has a different picture. So this is, I think, either part of or adjacent to the silver bog. So these are virtual horses. I thought that was a nice image. I actually walked around at the time and I took image screenshots, just as like a travel log, and I found those from an old website, and I put them on this one. So that was fortuitous in my case. Well, we have this is Richard's talk on January 9th of 2013, and in this case he has a reference list. So the reference list is hosted locally as well. So if you go to the reference list, it's just like references of academic papers that are noted in the abstract with these brackets. So 1, 2, 3, 4, 5. So there are a number of references, and those references are in this list. Here we go. So then the references are here. Probably need more work on like linking them to digital versions. So some of these are going to be available online, some are not. But that's the way it's presented. So I mean, right now it's set up like that, and it's pretty static. I'm not sure what could be added. I think that maybe there's some additional things that could be added to this, and maybe not directly related to the course. Like for example, like some sort of library of maybe agent-based models or something where people can do basic simulations of developmental stuff. Like different processes that they read about in these lectures. I mean, we have movies, we have other types of aids, links to external blog posts, but it's, maybe missing something like, and, something you can run in your browser. It doesn't have to be something really complicated. I used to have a list of different physics models that I found in, like back when they had Flash movies and Flash demos. I used to have a huge list of Flash demos for different physical models, like avalanche models and things like that. So I don't know if they have those as much anymore. I'll have to go look, but there might be some simple models that people can run on their browser like that would illustrate some of the concepts, walk through. It's easy when you see something running in your browser, some like model. It's maybe easier to understand the process. So I don't know. Yeah, I don't know. There's probably more to add to this, but right now it's pretty much the archival stuff is pretty much done. So, did you have any thoughts on that, Dick? Okay, add. So this is the conception and development of the Second Life Embryo Physics course. Okay, so there's a citation here. Okay, I'll put that on the. Let me see if I can add it now. Actually, I'll just add it into this for right now. And I can change this later, but I'm just going to add it in as a citation here. And so that's the paper, I guess, that they they wrote about this course. I'd forgotten about that, actually. I remember seeing it, I think, once or twice during the course, but that was a long time ago. So yeah, there's a lot of content there. Revisiting it, I was reminded of a lot of things that you generate an unknown list of the talks when you're done. Yeah, that would be good. I think that would be good to have like a reference, like or something like that, where you have like an endnote list is like a formal sort of set of citations for it. That would be good. With abstracts all searchable, yeah. Yeah, probably need a search function. So yeah, let me see. Yeah, I think that would be good. So I'll let you know when I'm done with it, and we can do that. I'll send you I'll just send you a link to the repo, and you can go through. Okay, we've got 2 Jessies now. Oh, we have 1 Jessie again. Okay. All right. Well, that's good. Yeah, I just wanted to update the progress on that. So, yeah. Okay. Any questions at this point? Anything anyone wants to bring up? I tend to talk a lot in these meetings, so I just wanted to give other people a chance to. Okay. So yeah, next week. All right. So next week we're going to have. I'm going to give. Okay. Ujjwal says I will have to go through the repo. I'll let you know if I have any doubts over Slack. That's good. And then okay, I'd give an update about the conference, but maybe more so next week. Good. That's Jesse. So for the next couple weeks. So next week I'm going to give a practice run of the talk I'm going to give to the virtual worm group. And so this is a group that was born of like this the lockdown. You know, people are not in their labs and they're at home, and a bunch of C. elegans people. Okay, I could talk on polygonal archaea and diatoms. Yeah, that's good. We can do this next week. The polygonal archaea and diatom stuff. I forgot about that. But yeah, that would be good for next week. So next week I have for 30 minutes I have this dry run of this talk, and so the last 30 minutes or the first 30 minutes, depending on how we go in order, Dick can present on the diatoms, and then Jesse said that her papers I've mentioned, yeah, in the future. So yeah, so next week then we'll do Dick's diatom talk, and then we'll do this virtual worm talk, and that was born from people who were affected by the lockdown. They're giving virtual talks in Zoom. It's kind of a private group, and each week like they have 1 or 2 people giving talks on different aspects of C. elegans biology. So it's really been very biologically oriented. There aren't a lot of computational people in the group. There are a lot of people doing different things with C. elegans, like, just basic biology type research. So, C. elegans is a model organism. But they do a lot of basic biology on C. elegans. looking at osmotic stress and sensory systems and other types of things. There was a wonderful talk last week on tracking behavior. But on the 6th of May, there's going to be 2 talks, and I'm going to give one. It's the computational week. So I'm going to go through those slides next week, and that'll be available afterwards on YouTube. And then the following week after that, we have the GSOC students. So the GSOC selection is next week. I think May 4th is when the successful candidates are announced. So then the week after that, I will have the people who were selected give a talk in here on their, like an introductory talk on their proposals, what they plan to do. And it's not going to be too formal because they only have a week to prepare, but hopefully we can welcome them into the group and we can ask some questions about what they plan to do. We already talked about the proposals in this group, so we will see, they'll get a little bit of interrogation by the group. So there's a community period in GSOC where people have to kind of get involved in the community, as well they should. And in previous years, we've done it separately from this group, so I want to kind of bring it together with this group more. All right, so so that's good. I think we've got that set up. So for the schedule, all right. So then the rest of the meeting, I think, we can go over the major tasks again, and then I want to maybe present a few papers that we have again in the reading queue. So let me see if I can get this up. Share my screen again. Okay. So the major tasks, again, like we finished a number of these. We've got actually quite an impressive list of finished things here. In progress, we have a number of things as well. Some things are on hold. And then to do, there's some other things that I haven't classified. I just wanted to go through the in-progress ones to see where we are. So the axolotl embryo animations and segmentation, that's something that I haven't really gotten to yet. I did the animated GIFs. I presented those a couple weeks ago. I haven't really gotten farther than that too much. I'm still thinking through, however, the technical details of that. Maybe we can open that up more in coming weeks. But I think I have a feel for the data at least, so we can work on that. Present on multi-cell systems. So this was something that Dick, when he talked about the polygonal bacteria or the diatoms in polygonal life, something that brought to mind this as well. But this is something that I can do in the next few weeks, maybe after Dick's talk, maybe the week after that, along with the GSOC students. We can just kind of quickly go over that. I wasn't really ready for that today. Create a doc for open collaboration of papers. So this is actually something that I don't know if I have a link to in here. I don't have one in here. Okay. I'm creating a doc for people to open collaborate on papers. So there's a number of papers that I have that we've kind of talked about, like abstracts and things like that, that we don't have. Like it can kind of get lost in the shuffle if we just mention it and let it go. So I'm actually creating something on GitHub, and I can't remember if I have it. I can't remember exactly where it is right now, but I'll find it. I think it may actually be in group meetings. So maybe we can go to that after this. But I've created a document that has kind of links to the Google Docs for these abstracts, for these open abstracts, so that anyone in the group can contribute. And of course, you have to ask for permissions to get in and do any sort of editing. But once you have the permission, then you can contribute in some way. I think there are also some. I also have lists of some GitHub repositories that are shared, so that's a good place for people to see, like what kinds of things we have open in terms of collectively written documents. Maybe they can contribute something like a paragraph or some sort of set of references. Oh, the collaborative project. So that it's basically, we've mentioned a couple of abstracts in the like for the Basel area cognition, and there are a couple others that we mentioned that were like open documents where we would have people from the group contribute different things. And so I just simply made a list of those. And I'm making a list as we go through so that if we have something that's collaborative, potentially people can, contribute. So you can either contribute through GitHub on some of them, which is like issuing a pull request saying this is what I want to contribute, or you would request permissions to a Google Doc and say, I want to add something into it. And, to make sure that someone doesn't, break, someone who isn't supposed to be there breaks in and erases everything or, puts a bunch of junk in the text. We want to keep that a little bit, have some securities on it. But, if Jesse, if you're interested in that, we can, I can, when I find the list, I can send it out and then you can look it over. And there are a number of different papers that are sort of outstanding in that way. Another presentation, this is still, I'm working on this. Make Google Summer of Code selections. That's going to be this, selections have already been made and I can't see anything about it. But we'll know the results of this on the 4th. So next Monday, we'll know this, the results. And I talked to Vinay about this, and we're going to promote this on social media. So when the selections are made, we'll issue some sort of mention of the people, and we'll get them some with an open room. Definitely, we're going to get them some exposure, letting people know these are new students for GSOC, and then maybe go over their presentation like in the following week, and we'll talk about that. So I want to be able to give them some exposure to the broader community of people who are interested in this stuff. And then, so the CompuCell model for C. elegans, that's Ujjwal, and we talked about that today. So that's continually in progress. Okay. Oh, the Synod stuff. Oh, yeah, I forgot about Synod. Yeah, I'll look into that. Yeah, there's a... Thanks for that reference. I mean, remember we were talking about Synod several years ago with respect to the cybernetic embryo. So that was... and Edmund Synod is... he's published a lot of books in different topics. So I'll look into that. Thanks. That was the one. That was the. Let's see. That was this issue here. Where is this? This one here, which I kind of glossed over, but I'm working on this thing with Moyer patterns, and then with like morphogenesis and perception. It's an agent-based model, but I'm not going to talk about it today. It's kind of an interesting thing. I'll present it in the next few weeks. But thanks for. I did need some references for it. That was actually something I was lacking in that. Schedule presentations. So we just did that. Nick volunteered to present next week, so if people are interested in presenting on things, please let me know. I can schedule it on our schedule. The bacillary non-neuronal cognition paper, that's one of those papers I talked about that were open, so we'll revisit that at some point in the near future. The Embryo Physics archive, so this is, I just showed this off. This is in progress, definitely. And then reviewing the axolotl data. So this is of course, this is being reviewed. So I guess this is done, but I need to make another issue about working with the axolotl data. So I think that's all for the in progress issues. I just wanted to see if I could find that list of let's see, group meetings. Okay, this is it right here. So it's the README and group meetings. Let me send a link of this put a link of this in the chat. Okay. And so this is wait a minute. This is the file that I was talking about. So this is just the README. So we have links to the YouTube channel and the weekly agenda. This is actually where I put the schedule for coming weeks. It's some Excel file in Google Doc, and I just enter in people's names, pencil them in, or type them in, and then revise it as necessary. So these are the documents that we've got. Some of these are things that I've kind of been working on. Some of these are like more, established group projects. But none of them are very developed, and it's just, people are interested in contributing to them. You can go through and look at the documents. Sometimes they're very rudimentary, but sometimes they're actually, actual outlines. Like for this one's an outline. This one is actually, this was based on the lecture that I delivered at Neuromatch, but it might have some relevance to this group, and I put a GitHub repo link here. So if you want to contribute to the GitHub repo, this is the GitHub repo for this particular idea. And so there's a lot of stuff in here. If something strikes your fancy, you can contribute to it. And you don't have to issue a pull request. You can just email me and say, this is something that relates to this thing. I try to keep this open as much as possible in terms of multiple channels, because we have some people who are very proficient at GitHub and other people who don't use it. So if that's that, that's where you would go for that. And then I guess the last thing I'd like to do today is to go through some papers that I found in the reading queue. There are actually 2 papers this week. Last week I went through two very, I guess you could call them hard papers, because they had a lot of content in them and they weren't immediately obvious what they were trying to do in the paper. We had some technical difficulties, and I said that I would prepare some slides on the second paper, but I didn't get to that yet. But it's this week, these papers are a bit less challenging, although they are pretty, they're pretty technically involved. So I'm just going to go through them very quickly, just to make you aware that they exist. You don't have to read them all the way through if you don't want to. This is something that Steve McGrew pointed me to this last week. He sent an email, and the story, again, like last week I said that sometimes like these press releases that they give for papers are particularly misleading, or they're very hyped. And then when you read the paper, it's something different, but it's still impressive. This one falls into the still impressive category. It says Morphoseek. So this is where they took a chordate. In this case, it was a C-squirt, and they looked at the cells in the early embryo. So one of the things about the early embryo sea squirts is that they have this very orderly division process. So they actually have been able to very exquisitely characterize the phenotype. Each cell divides into daughter cells, and those daughter cells are easily identified in the embryo. You can track them, and they're always the same. It's like C. elegans. But in these embryos, instead of having a simple left-right symmetry, they have a fourfold symmetry. So they actually have 4 equivalent hemispheres. And so that has something to do with the way that the cells divide. And the way the nomenclature works is it's like the nomenclature is like the cells divide into A cells and B cells, and then there's an A star and a B star. And that's how you represent that fourfold symmetry. But the advantage of this type of development is that you can identify cells and track them as they go through this early embryogenesis. So what you can do is you can attach gene expression data to each of these cells, and that's what they do in this paper. They do this thing called Morphoseq, which is single-cell RNA sequencing. And so this is a specific type of RNA sequencing. If you're familiar with what they call next-gen sequencing, you know that there are a lot of different methods for taking RNA and sequencing it. But this one is based on recovering RNA from a single cell, amplifying that RNA, and then sequencing it, and then mapping it to some reference genome. But in this case, they're also taking it and mapping it to that cell. And so that cell and its cell lineage, so every daughter cell, they have these data. So now they can actually trace through what they call the lineage tree and identify single cells by their RNA profile. And so they've done some of this with C. elegans, but they actually haven't done it in development so much. They've done it in adults, and they have atlases where you can look at adult cells and you can get the gene expression profile for adult cells. But for development, it's been a little bit elusive. And so they describe this technique here. And so but the additional thing that they've done in this work, and this is an example of how the cell divides into this fourfold symmetry, right? And then eventually you get tissues and things like that differentiate. But the additional thing they've been able to do in this paper is to build a 3D model, a 3D computational model, and predict the cell division process. So I don't know if they have it in here. I don't know if they have it in the paper. But they have it in the article. Okay, so this is, I think, an example of how they've done this. So they can actually map these gene expression profiles to a virtual model of the embryo. So in this case, you have this cluster of like 3D spheres, right? And they've been able to actually map these to specific cells in this virtual model. So this is just a model of what you observe in the embryo. And most of the time when you do RNA analysis, you're maybe able to localize it to certain parts of the embryo or certain cells, maybe. So it's very kind of approximate. In this case, they've been able to actually map them to single cells. So they can define single cells by defining their RNA expression profile. And again, this wouldn't be possible necessarily in a mammalian embryo because, it may be possible to identify cells, but it wouldn't be the same in every embryo. In this case, you can do this much easier in this type of embryo. And so this is another example of the virtual embryo that they've used. Vegetal pole. This is an example of the vegetal pole from this embryo, and they have different cells that have protocadherin expression. So, for example, this is where these are the expression patterns for that. So that's a pretty interesting paper. Let me actually give you a link to this folder in case you want to download these papers and read them yourself. So that's the paper on MorphoSeq. And then in the last couple minutes, I'll talk about the second paper, which is post-Turing tissue pattern formation. So this is something, so the Turing model, of course, is the reaction diffusion model that we know and love. And it involves, it's sort of a chemical morphogenesis model. In this case, they're advocating for this mechanochemistry model. And so the abstract says, chemical and mechanical pattern formation is fundamental during embryogenesis and tissue development. Yet these underlying mechanisms are still elusive. Most current theories assume that tissue development is driven by chemical processes, either as a sequence of chemical patterns, each depending on the previous one, or by patterns spontaneously arising from specific interactions. So that's your Turing model. Within both theories, mechanical patterns are usually regarded as passive byproducts of chemical pre-patterns. However, several experiments question these theories, and an increasing number show that tissue mechanics can actively influence chemical patterns during development. So their model focuses on that interplay of chemical and mechanical patterns. And so this is, let's see if they have any figures in here. They kind of talk about the Turing model and how it's inadequate. And then here's a figure 1 way down here. Okay, so this is the example that they give of how this works. So there's a feedback loop between tissue mechanics and morphogen production. So you have this morphogen production going on here at the surface of the cell, and it emits this morphogen. Morphogen is theoretical, but in real life, it's some sort of chemical signal that tells other cells, how to deform along with the cell that's emitting this morphogen. So, it's a collective behavior thing among cells. But then there's also this elastic response. So when you have, say, for example, a bunch of cells in a tissue, they might stretch in some way or they might contract in some way, and that's a signal as well. And they're well-known mechanical transduction pathways in cells that allow them to respond. And so they're kind of fitting this into one theory. So Dick says, Turing discussed this in his paper. Yeah. I mean, it's not like people didn't know about this. I guess in this paper they're trying to really stitch it together. They do mention this up at the top. So I don't know what their actual beef with. I don't think there's a beef with Turing necessarily, but okay, they actually do distinguish it from Turing theory. So the Turing theory requires highly nonlinear interactions among different types of morphogens in order to produce de novo patterns, which makes the underlying assumptions regarding molecular interactions relatively complex. So I guess their argument is they're simplifying this relationship. I talked about this with someone in the area of neuroembryoids, where they're creating these embryonic structures of neurons, and the question is how do they differentiate into neurons, and how do they form a coherent neural system? And we were talking about like the standard model of looking at connections, but also about the role of mechanobiology and things like that. So this has a lot of this sort of this model, this type of model where you're mixing in chemical morphogenesis and mechanical morphogenesis is pretty relevant to a lot of different areas, not just embryos. Let's see. So yeah, they do these really high rent simulations here, where they have they look at morphogen contraction. They look at the simple mechanochemical feedback loop, so they're actually able to model the interaction between the two things. And these are numeric simulations. Then they vary the morphogen concentration across the sphere that they're modeling, and they can make different shapes using the same feedback loop. In this case, they're looking at something called apical constriction. So they're looking at a specific type, thing that happens in morphogenesis. And so this is an interesting paper. I don't know how much ground they actually break in this paper, but it's I found it pretty interesting. I thought it was worth bringing the group's attention to. Again, like I said, there's a lot of detail in here I'm not going to go through, but it's something that you can read on your own if you're interested, or if you have further questions about it, we can talk about it. So that's all I have for today. If that's all everyone else has, we're at the top of the hour. So does anyone else have anything that they'd like to mention before we go? Yeah, thanks for attending. Okay. Yeah. Stay safe as well, Ujjwal. Everyone have a good week. Okay. Jesse said, I might ask you about GI papers in developmental biology, but I'll say that in Slack mostly. Key. Okay. Yeah, we can do this in Slack. So, talk about this in Slack or by email if you have further things to follow up on. So, okay. Thanks for attending. See you guys next week. Bye.